Dear students, today we are going to discuss about kingdom Monera, which includes prokaryotic organisms. Now, one very important feature about the prokaryotes, they always are unicellular, and they never found into the form of multicellular organisms. So you are supposed to keep this into the mind that prokaryotes or Monera are always unicellular. Now, few more important characteristic feature if we see, they are most abundant microorganisms. Now, most abundant in the sense of number, not into the variety, right? The types of bacteria and number of bacteria, they are totally different phenomena. So, most abundant word is used because they are found in huge number whensoever and wheresoever they are present. For example, hundreds of bacteria are present in only a single handful of the soil. So there are so many, or they are huge in the number. The other important characteristic feature is they also live in very extreme habitats. For example, in hot springs, in deserts, snow, even under deep oceans, and many of them are parasites. So these are the few common characteristic features of the monerans. Please take the screenshot so that we can move ahead into the next slide. Fine. Now comes the categories or types of the bacteria. So if we see here, we can find 4 types of bacteria around us, right? So it is called types based on shape. Right? They are the types of the bacteria which is based on shape. Number 1, they are spherical in shape, and it is called coccus. Number 2, they are rod shaped, and it is known as bacillus. Number 3, they are coma shaped, and they are known as vibrio. And 4th, they are spiral, known as the spirillum. So these are 4 various types of bacteria which is based on the shape. So bacillus, coccus, vibrio and spirillum, these are the 4 types of the bacteria found around us. Please take the screenshot. Now, if you see the diagram which is given here, you can find internal and external structure of the bacteria. Now, according to the structure of the bacteria, it is very simple in structure, but they are very complex in behavior. Now, though they are very simple in structure, but they can survive into varieties of the condition, varieties of the atmosphere where other organism cannot. That is why it is said that they are complex in behavior. They are found in extreme conditions where normal organisms could not be able to even survive. A reason behind that is they have metabolic diversity. They have special type of mechanism of the metabolic reaction, and that is why they can survive easily into the extreme condition. So that's very important feature they have, right? Some bacteria are autotrophic, means they synthesize their own food from inorganic substances. So they absorb so many different types of inorganic substances from their surrounding, and they form organic compounds within their body. And majority of them are heterotrophs, which are unable to synthesize their own food. So they do not synthesize their food, but depending on the other organism or dead organism. So either other organism or dead organism for their food. So this is the structure of the microorganism or bacteria. So few more characteristic feature respect to their structure, what we are about to see into the next slide. So before that, please take this screenshot of the slide. Now this is the classification of the bacteria. Bacteria are of two types. Number one is Archaebacteria, and the other type is Eubacteria. Archaebacteria are of three types. Number one, they are halophiles. Number two, they are thermoacidophils. And number three, they are methanogens. Eubacteria are of two types: autotrophs, which may be either photosynthetic or chemosynthetic. Photosynthetic, they are preparing their own food with the help of the sunlight, and chemosynthetic, they prepare their own food with respect by absorbing various types of chemical from their surrounding. And the second type is heterotrophs, parasitic or saprophytic. When the microorganisms live either on or within a particular other living organism, they are parasitic, and saprophytic, they depend on dead or decaying substances, they are saprophytic. So these are the various different types of the bacteria which are found around us. Now Archaebacteria and Eubacteria, we know that basic difference between them is Archaebacteria survives under the extreme condition, whereas Eubacteria are found in general habitats. So that is the difference between both of them. Please take the screenshot so that we can move ahead. Now this is the story of the Archaebacteria. There are three basic types as we have seen earlier: halophiles, thermoacidophils and methanogens. So as we know that Archaebacteria live around very harsh condition, right? Very harsh condition. For example, if they are surviving in extreme salty habitat, they are halophiles. If they found in hot springs, they are thermoacidophils. Or they found in marshy area, they are methanogens. Methanogens produce methane gas, right? That's why they are methanogens. Archaebacteria have different cell wall structure which are protected. So they survive into the extreme condition. It is protective cell wall. It is the adaptation, because whensoever onto the earth very primitive, very first type of organism, they were Archaebacteria, and at that time when they have started to grow on the earth for the first time as a living organism, the atmosphere of earth was very extreme, very harsh. So to protect themselves, they have created this special type of adaptive mechanism of the cell wall. Right. So that also you are supposed to remember. Methanogens are present into the gut of ruminant animals, for example cows, buffaloes, etc. And that's why they are very important in the production of biogas from the cow dung, right? Of this, why cow dung of these animals are important in biogas? This is the reason which is specified here, right? So they have methanogens. So that's the importance, economical importance of methanogens right now. So that's the story of three different types of the Archaebacteria which are found in the extreme condition. Please take the screenshot. Now about Eubacteria. Now they have very rigid cell wall and flagellum around the surface of the body. Now flagellum are not found in the bacteria which are immotile, that is cannot move from one place to the other. So if they are motile, that is moving from one place to the other by swimming, then they must have flagella, and flagella found to be locomotory component or part of the organism. They have basically of two type. Number one, autotroph, either photosynthetic or chemosynthetic, and other type is autotrophs, heterotrophs, which are depending on the other for their food. So autotroph either photosynthesizing or chemosynthesizing, or they are heterotrophs. So these are the two basic type of the organisms. Please take the screenshot. Now let us first talk about the photosynthetic autotrophs. For example, they are also termed as the Cyanobacteria, right? Please do remember the extra name as well. So Cyanobacteria are photosynthetic autotrophs. So they have chlorophyll A, right? They have chlorophyll A, and that chlorophyll A is pigment which is responsible for photosynthesis, which is similar to found in green plants as well, and that is why they are also termed as Cyanobacteria, that is blue green algae. This is the third name. So photosynthetic autotrophs, for example Cyanobacteria or blue green algae, they are either Cyanobacteria are often found in the water bodies around us. Ready? So you can have the example like where they are found. So they are generally found on or within the water bodies around our surrounding. Now they are, number one, unicellular, so it is has to be because they are Monera. Colonial, found in a group, maybe filamentous, thread-like structure they form, right? So when they are spherical in shape, when they are in the group, they are colonial, and when they are thread-like structure, forming thread-like structure and living together, then they are filamentous. So this is the example of filamentous, right? So they can be marine or terrestrial algae. So freshwater or salty water, in both the medium they can found. The colonies are generally surrounded by gelatinous sheath around the structure of the body. So very special protective sheath which is slightly gelatinous, contains gelatin, right? So due to that, they are, it is gelatinous means it is very sticky substance around us, so they can join together to form either colonial form or filamentous form. So gelatinous structure or layer makes it possible. Ready? So they often form blooms into the polluted water body. Very large blooms, they are found on the water which are getting polluted. Now some of them are fixing the atmospheric nitrogens, right, in a specialized cell called heterocyst. This is heterocyst which is present here into the diagram. Ready? It is the heterocyst into the anabina. So into those colony or filament, a few cells are there which are heterocytes, and they are fixing the nitrogen into them. And two basic examples of the Cyanobacteria are nostoc and anabina, which is given into the textbook. So please take the screenshot of the slide. Now second is chemosynthetic bacteria. Now as the name is indicating, chemo, that is chemical, so they utilize various types of chemical substances from their surrounding and they synthesize their own food. So chemosynthetic autotrophs are oxidizing few inorganic substrate, for example nitrates, nitrites or ammonia. They absorb either nitrite or ammonia and use them for releasing energy for their ATP production. So they are using these components as their food, right? And they produces their energy, and that's why they are chemosynthetic. Now they play great role into the recycling nutrients like nitrogen, phosphorus, iron and sulfur. Basically they are found in soil. So dead decaying material and waste product which are added to the soil, they are getting composed by, so they are, sorry, not composed but decayed by this kind of autotrophs, chemosynthetic bacteria. They are also found under the ocean, very deep ocean where sunlight is not possible to reach. There they found, and that's why they are supposed to be chemosynthetic, because they don't find sunlight there, so they cannot be photosynthesized, right? Please take the screenshot. Now comes the third type, heterotrophic bacteria. Heterotrophic bacteria are most abundant because they are found in huge in the number, most abundant, and majority of them are important decomposers. So they found as a decomposer. Now what is decomposer? Whatsoever dead and decaying material which are present on the earth has to be decayed, converted back into the inorganic substance from the organic, and that is the important role which is played by these decomposers which are present in the soil. So they are in the soil and they are decaying the dead substances. They are decomposing the dead material or decaying material. That's what the important, very important role with respect to the balance into the ecosystem, right? So please take the screenshot. These are few examples of the heterotrophic bacteria and few characteristic features as we have seen into this slide. The impact of the heterotrophic bacteria on human affair, how they are beneficial to humans. Number one, they are used to make curd from milk, as we know. Number two, production of antibiotics. We produce so many different types of antibiotics with the help of the bacteria. These are the drugs what we are used to cure or recover from the bacterial infections. 3, we also take benefit of fixing nitrogen from leguminous root. Leguminous are the name of the or types of the plants, legumes, which produces the pulses. Number 4, some are pathogens, so they are harmful, causing damage to the human being. So 1, 2, 3 are useful bacteria and 4th is harmful bacteria. So damage to the human beings, crops, farm animals and pets. For example, they can cause cholera, typhoid, tetanus and citrus canker. So these are the few impacts, maybe positive, maybe negative, into the human life which is caused by these kind of bacteria. So please take the screenshot of this slide. Now if I talk about reproduction method of the bacteria, the main reproductive method is fission. Now fission is simply divide from 2 from into 2 from 1. So if one cell is dividing into 2 is simple fission. Ready? Under unfavorable condition, they form spore. Now what is spore formation is given into the diagram there. So normal vegetative cells, they are dividing by dividing internally, if you see into the diagram here. So it is dividing internally. The one half is denatured automatically. So only one part is left here inside the cell. This is cell wall. This is cell membrane. So cell membrane is converted into coat, right? It is developing as a coating, special coating, multi-layered. It is converted from one layer to multi-layer, and that multi-layer structure is the spore. So that is how spore formation is found, and that multi-layer structure is giving specialized protection under unfavorable condition, and that is the only why it is the adaptive method under the extreme or harsh condition for survival of those kind of bacteria. They also reproduce by sort of asexual reproduction by adopting the primitive types of the DNA transfer from one bacterium to the other. It is called fusion, right? Method is called fusion. Please take the screenshot of the slide. Now the example which is given here is mycoplasma. Now what is mycoplasma? It is to be remembered, it is the smallest living cells known within the living organism. Though it is a bacteria, it is a eubacteria, it is especially adapted. Do remember that. This is the organism which is found without cell wall. Though they are bacteria, they are without cell, right? So it is without cell wall, sorry, not cell. General characteristic feature of the bacteria is they contain cell wall, ready? But they have lost the cell wall by course of time. So they are specially adapted, and they are without cell wall and smallest living organism which are found within the earth. They can survive even without oxygen, right? So they don't need oxygen, anaerobic it is said to be, right? So they survive under the condition where they don't have oxygen around them, and many of them are pathogenic either in humans or plants. They are disease causing microbes. So they are the smallest living organisms. First time when they are found, it used to known as PPLO, pleuropneumonia like organisms. But as researches have were going on, and furthermore classification and furthermore the characteristic feature have been found, it is converted, or it is known to be now as the mycoplasma. So maybe in few reference books you also find PPLO word. So PPLO matlab pleuropneumonia like organism, and right now they are known as mycoplasmas. So don't get confused with those words, please. Fine. I'm ending my session right now with the Monera, and we'll be continuing with Protista into the next lecture. So till then, I'll see